Today's lecture is about cDNA library. Chromosome DNA and genes are present inside the cell nucleus. So inside the nucleus we see chromosomes. These chromosomes are nothing but highly condensed form of DNA along with histone proteins. And this DNA double helix has various regions. Some of these regions constitute of the genes. DNA consists of non-coding and coding regions. 2% of the entire human DNA codes for proteins, whereas the 98% is the non-coding DNA. These non-coding DNA have regulatory functions and various other functions, whereas the coding DNA is the part that makes proteins which totally regulate our body. So this is the human DNA having coding regions which are also known as exons and the non-coding regions which are known as introns. Exons have the genes in them. It is these genes that make proteins and the proteins regulate our body. What comes to your mind when you first hear the word library? Yeah, so library is a collection of well organized books. Similarly, gene library is a collection of different fragments of our DNA. DNA library are of two types, that is genomic DNA library and cDNA library. Genomic DNA library is a collection of cloned DNA segments that represents an entire genome of an organism. Enzyme used is reverse transcriptase. The library consists of both exons as well as introns. So information in genomic DNA library is huge and complex. The vectors used for constructing DNA library are BAC and YAC, as they can carry large DNA fragments for cloning. The cDNA library, on the other hand, contains only expressed DNA. It is a collection of cDNA produced from mRNA that is present in the nucleus. Enzyme used is Restriction Endonuclease. The library contains only exons. It has less sequence information. For example, at a given point in time, of the 26,000 genes present in humans, only 20,000 genes are expressed. Then the information to be screened is that much less. Vectors used are plasmids and phagmid, as they can carry small cDNA fragments. Now we will take a look at the building of cDNA library step by step. The mRNA is first extracted from a specific tissue. It is used as a template for the synthesis of cDNA strand. The enzyme used here is the reverse transcriptase enzyme, which can synthesize DNA from mRNA. A single-stranded cDNA is obtained by using reverse transcriptase enzyme and an oligo-DT primer. The second strand of cDNA is synthesized with the help of enzyme DNA polymerase, and then the latter is transferred into a cloning vector. The vector is either a plasmid, phagmid or lambda phage. This vector is transferred to a recombinant host. The host is generally E. coli. When the E. coli multiplies, this recombinant vector also multiplies, and there is a collection of clones of cDNA, and this is known as cDNA library. Genomic library therefore refers to a large collection of bacterial cells. As you can see here, these are the large collection of bacterial cells, each containing a random piece of human genomic DNA which is found in the Petri plate. cDNA library, on the other hand, is a collection of all the expressed DNA of a particular cell type or tissue. For example, A cDNA from pancreatic beta cell contains clones with cDNA for proinsulin, and a cDNA library from bone marrow will contain many clones with cDNA for alpha and beta globin chains of hemoglobin. So cDNA library is dependent on the tissue of origin. It is present in the Petri plate. Thank you for watching. Like, comment and share and subscribe to my channel.